I realize my talk is a little bit apart from the molecular mechanisms which we heard about in the previous talks, and I need to switch gears back to cellular neurophysiology, which started discussed in a talk by Professor Zudov, and yes, okay. So it now works, and I would like to discuss work which I was doing for maybe last 15 years, and it's related to activation of extrasynaptic GABA receptors, and this is very interesting in the context of extrasynaptic signaling in general, which my lab is working on. And I would like to start my presentation from this image, which is very known to most of neurophysiologists, and this concept was nicely introduced in the talk of Professor Zudov. This concept is a synaptic organization of the brain. So this schematic comes from one presentation to another, from one textbook to another, and basically the main idea here is brain is a large synaptic network. So neurons, electrically excitable cells, they're connected to each other by plastic synapses, and these synapses might appear and disappear, making rewiring of this network, and new neurons can also appear and get introduced into this network, and this is basically modern idea about how the brain works and how the information is stored and processed in the brain. So again, from the discussion from Tom Zudov, in the United States, about 80% of the labs working on the neural circuits and synaptic signaling, synaptic organization of the brain. And today I would like to introduce another side of this story, another side of this research on the cellular neurophysiologist, and I want to point one issue in this image. So there is no such empty space between neurons and the brain. Of course, it might be filled by unnecessary cells, but you see, if you look at the electron microscopy, you will find out that all the extracellular space is filled up with other neurons, astrocytes, blood vessels, etc. And so there is no such thing as extracellular space. It looks like rather extrasynaptic cleft. And in this extrasynaptic cleft, we also have changes in concentration of neurotransmitters, and we also have presence of extrasynaptic receptors there. And the role of this extrasynaptic neurotransmitter and extrasynaptic receptors is also very important. And today I would like to talk about one type of extrasynaptic receptors, inotropic GABA receptors. Inotropic GABA receptors are pentameric structures, so this receptor consists of 5 subunits, and at the moment I think identified more than 18 types of such subunits, and which gives us a huge number of possible combinations. However, there are certain rules how receptors are built up and these subunits are assembled, and in reality we might be dealing with about 30 such subunits, and actually majority of these receptors have similar structure. It's 2 alpha, 2 beta, and 1 gamma subunit. This specifically concerns synaptic receptors. Extrasynaptic receptors might also have, instead of gamma, delta subunit, and there are other forms as well of these receptors. And receptors are brain region specific. They also cell type specific and might also have different subcellular distribution, and of course a synaptic and extrasynaptic distribution too. And this large variety of receptors and large variety of sources of GABA also prompts different types of GABAergic signaling. The classic one is synaptic GABAergic signaling, illustrated here in this picture. So it works in a classical way. Presynaptic terminal triggers release of vesicle with GABA, and GABA activates postsynaptic receptors, and which produces fast transient, which we call IPSC, inhibitory postsynaptic current, or IPSP, depending on what we're measuring. In the terms of this talk, it could be called phasic response, phasic current. Extrasynaptic GABA receptors, they sense a small concentration of extrasynaptic GABA, and they have different properties. They have slower kinetics of desensitization and inactivation, so they can, once they bind to GABA, they can produce much longer currents. And therefore, when many extrasynaptic GABA receptors activated, we can detect persistent tonic current. And this you can see here. And nowadays, many labs working on this extrasynaptic receptor-mediated tonic current. However, the whole story is relatively recent, and particular interest from many labs probably started about 20 years ago, when detailed description of tonic current was published, tonic current mediated by GABA receptors. And the favorite subject for this research is hippocampal slices or brain slices in general. The reason for that, then, if you work with cultures, so obviously you lose three-dimensional structure of the brain, and in cultures you cannot have all this extrasynaptic concentration of neurotransmitter. So the network which is built in cell cultures is lacking basically extrasynaptic receptor activation. And slices have advantage over in vivo work in terms we can easily access our cells. We can, first of all, we can visually identify them. We can easily access them with patch electrodes. And we also can control many parameters of our recordings, for example, drug concentrations. So this is a big advantage, but of course disadvantage of the slices that they lose all the afferentation, all the input and output, which characteristic for this network in the intact brain or in vivo. Therefore, it's very important to somehow translate the experimental results obtained in slices for in vivo work, in vivo recordings. So how we do these recordings is illustrated here. So we recorded in our experiments either from excitatory pyramidal neurons in CN1 region of the hippocampus or from inhibitory interneurons. And so you can see on the left schematics of our experiment, and in the middle you can see DIC image. This is infrared differential interferential contrast image. You can see interneuron in stratum radiatum of the hippocampal CA1. We've attached or we've connected a glass pipette on it. Then we keep cell membrane in voltage clamp and recording current. And when we do whole cell experiments, we're not only clamping voltage on the membrane, but we also clamping ionic concentrations. And when we record with high chloride concentration side of the pipette, we quickly replace the chloride ions in the cell, and therefore we change reversal for potential for GABA currents, and GABA become depolarizing, and we look at the currents entering the cell. And if you block glutamate receptors, AMPA and MDA receptors, if you block metabotropic GABA B receptors, so we can see this kind of spontaneous responses, which we call spontaneous iPSCs. We know that these are spontaneous iPSCs because they can be completely blocked by antagonist of GABA receptors, picrotoxin. And that's what you can see here when we apply 100 micromole of picrotoxin. These spontaneous iPSCs are gone. But in addition to the blockade of spontaneous iPSCs, one can detect shift in holding current. And this shift in holding current is exactly tonic activation of GABA receptors because it's present there and it's persistent, unlike phasic iPSCs. So this tonic current can be measured, and response to different manipulations can be obtained. And already in pioneering works in the field, people describe that tonic current is cell type specific phenomenon. So here on the left, you can see our recordings done in hippocampal interneurons and pyramidal neurons, where we found that tonic current is really large in interneurons and almost absent in pyramidal cells. Similar data were published on neurons in thalamus, when in different nuclei in some cells tonic current was present, in other it was absent, and later on more papers came about other brain regions. One simplest explanation for the type cell specificity of tonic current would be, of course, distribution of GABA receptors. So different neurons express different GABA receptors, therefore some express tonic current, some don't. But again, we looked at the other possibility. We looked at that cells might express GABA, extrasynaptic GABA receptors, but these receptors don't simply see GABA. We look at the possibility that GABA may not be uniformly distributed in the brain. And this idea was hinted basically by distribution of GAT1. This is GABA transporter 1. This is the major transporter that takes up synaptically released GABA in the hippocampus. And we found that it's not distributed evenly. And you can see here that most of GAT1 is located in pyramidal cell layer, but almost absent in stratum radiatum, where we recorded from interneurons. And in pyramidal cell layer, we had most of the cell bodies of pyramidal neurons. Therefore, this could be explanation why we didn't see tonic current pyramidal neurons, because all GABA was taken around these cells. And we could see tonic current in interneurons because GABA was present, so there was no mechanism to remove it. And to test this, we applied NO711. This is the drug which blocks GAT1. And we found that NO711 increased tonic current in interneurons and produced huge tonic current in pyramidal neurons. And total amount of the current in the presence of GABA receptor antagonist was similar in interneurons and pyramidal cells, suggesting that in principle, interneurons and pyramidal cells both have extrasynaptic GABA receptors, but pyramidal neurons are shielded by GABA transporters, therefore cannot express tonic current because the GABA is not just reaching them. And this is another evidence how we confirm this finding. We both applied different amounts of GABA, and we found that 1 micromole of GABA produces a large response in interneurons. Of course, 5 and 50 micromole produced corresponding increase in response, a larger tonic current in interneurons, but not in pyramidal neurons. And this brings me to intermediate conclusion in my talk, which we made at the time when we published this work. And this conclusion is following. So let's imagine that interneurons exposed to ambient GABA and express tonic current. So they being kept under a certain level of inhibition all the time. And if GABA decreases by one other reason, it decreases tonic inhibition, tonic conductance in interneurons. And this in turn should have network effect by increasing interneuron excitability and synaptic inhibitory synaptic drive to principal neurons, to pyramidal neurons. But these days, I put this last suggestion in red, and I put a question mark, and I will explain you. So later on, we figured out that the situation is not that straightforward. So, and excitation inhibition are not necessarily correlate directly with the amount of tonic conductance. But I would like to quickly remind you, especially for students, molecular biologists, some basic principle what is excitation inhibition in the brain. Excitation is a process which increases probability of action potential generation, while inhibition is a process which decreases probability of action potential generation. And let's think of the simple excitatory event. It's excitatory postsynaptic potential, EPSP. What it does to the cell is depolarizes the cell, and cell membrane potential become closer to the threshold for action potential generation. At some point, EPSP can reach the threshold for action potential generation, then cell fires action potential. Very simple. But when we talk about inhibition, this is not that simple. What you probably remember about inhibition is hyperpolarization. If you hyperpolarize the cell, you bring the cell membrane potential further away from the threshold for action potential generation, and EPSP cannot reach anymore. For example, we had originally in this model made in neuron resting membrane potential minus 60 millivolts, and if you move it to minus 62, EPSP doesn't reach threshold anymore. So this is classical and most simple way how inhibition can be exerted. But another way how you can inhibit the cell, it's by changing the membrane conductance. So if you recall Ohm's law, so you can figure out easily that EPSP, excited postsynaptic potential, is equal to excited postsynaptic current divided by the membrane conductance. And if the membrane conductance increases, so you divide the current by larger value and you get smaller EPSP. Very, very simple. But how it works, if your, for example, reversal potential for your GABA current is equal to resting membrane potential, even more depolarizing, you still introduce additional conductance to the cell membrane. So more conductance, you will get more inhibition. So GABA can inhibit the cell not only through polarization, depolarization, or hyperpolarization, whatever happens to membrane potential, but through shunting. This effect of additional conductance on EPSP called shunting. So this is very important for my later explanation. And indeed, if you apply different concentration of GABA and measure membrane conductance in interneurons, how we did it here, so we looked at the current response to hyperpolarizing voltage step, we could see that addition of 1, 5, or 50 micromole of GABA produced increase in membrane conductance proportional or corresponding to the GABA added to the bath. And another feature of GABA-mediated signaling in interneurons in different brain regions is actually that GABA not always hyperpolarizing in these cells. Of course, all you know that GABA might be depolarizing in immature brain. But again, some people cast doubts on this. But when we work with adult brain, developed brain, so we see GABA hyperpolarizing in excitatory cells, but in inhibitory interneurons, we still can see GABA slightly depolarizing. Not very much, but more depolarizing at least than in excitatory cells. And this is very important. And we confirmed this for hippocampal interneurons. So we made IV curve in hippocampal interneurons for GABA-mediated current, tonic current, and we looked at and we measured the resting membrane potential. This was done in gramicide perforated patch experiments. So this is the way how these things can be measured. And we found that resting membrane potential was more hyperpolarizing than reversal potential for GABA. Or in other words, GABA was depolarizing in these cells. And this is very important, again, because interneurons spontaneously fire in cells. So they can generate action potential without additional excitatory input. And even small depolarization of the cell is enough to increase their firing rate. And so we looked how their firing rate changed under the influence of GABA, and we were completely surprised. What we found is biphasic effect of GABA on excitation of interneurons. So when we add small concentration of GABA, 1 micromolar, we can see increase in firing rate of interneurons. When we increase concentration of GABA to 5 or 50 micromolar, this excitation was suppressed and switched to inhibition, so firing was suppressed. And if you look at this data on frequency change of cell firing against the membrane conductance, you will see this biphasic effect. Small addition of GABA, is what we think, depolarize the cell and make interneurons to fire more frequently. However, when concentration of GABA increases, what we have more and more shunting effects, so inhibition will dominate. And again, coming back to the question which I raised at the first part of my talk, what happens if the tonic conductance in interneurons changes? So now I can tell it for sure. It's not simply increasing or decreasing synaptic input to pyramidal neurons. It has biphasic effect. Small doses of GABA, small amounts of tonic conductance excite interneurons, so increasing inhibitory drive to pyramidal neurons, and large doses basically suppress this inhibitory drive. And so this is a conclusion of my second part of this talk, and I'm not going to stop because I